Hi everybody, this is Rebecca at Betterbred, and today I'm doing a presentation on IR. The goal of this presentation is to familiarize yourself with the concept of IR. But before we get started, I'm going to go over some definitions that may be confusing for you. Now, these may take a little bit of time to familiarize yourself with, and if you are not already aware of these different definitions, then feel free to pause and read over. A gene is a distinct region of chromosome that is inherited from parent to offspring. An allele. It's a variant form of a gene. Some genes have a variety of different forms, which are located at the same position or genetic locus on a chromosome. For example, we have different genes for coat color in dogs, so black, brown. Those are two different alleles of a gene. Heterozygous, an individual containing two different alleles. So a dog that carries for black and for brown is heterozygous at that particular gene. It has two different alleles. Homozygous, an individual containing two identical alleles. So, for example, a dog that would have two genes for the color brown and therefore would be brown. STR, short tandem repeat. It's a unit of DNA repeats that happen over and over hundreds of times in a row. There can be many different types of STRs in dogs, so that's important to know. More explanations following. So here's a picture of chromosomes. This is a pair of chromosomes. In dogs, there are 39 chromosome pairs. Then here's an example right here. This is a locus or a gene. This is the location on the chromosome for a particular gene. Here is a homozygous chromosome pair. Here's heterozygous, heterozygous, and then homozygous. Now, it doesn't matter what order you put big B and little b in. I got that question at one point. And then these are different alleles. Big B and little b are different alleles, but it's at the same gene and locus. Short tandem repeats. Here's a visual. Those of you who watched the video on the UC Davis certificate will recognize this. This is one chromosome, this is another chromosome, and this is a heterozygous chromosome because it has two different types of repeats, which would be two different alleles. At each location, every breed has anywhere from 2 to 27 different variations of these repeats. These are STR markers, or the alleles that are reported. Again, this is a heterozygous dog, so the markers are different from one another, the alleles. And a homozygous dog would have, say, 5 repeats and 5 repeats. Now, I just said 5 and 6 repeats so that I didn't have to make more blocks, but it can be very many different repeats. Why STRs? STRs are commonly used to track genealogy and parentage. If you think about it, in breeds we want to increase or maintain diversity in different genetics. This means we need to identify different genetic ancestors. STRs are perfect to help identify dogs who still maintain different genetics from their ancestors. Internal relatedness. IR stands for internal relatedness and is a measure of genetic diversity within an individual. It takes into consideration both heterozygosity of alleles at each STR and their relative frequency in the population. Therefore, IR values heterozygosity over homozygosity. IR values are unique to each dog and cannot be compared between dogs. Two dogs can have the identical IR values but have very different genetic makeups. And this is very important. So you can have two dogs with, say, an IR that's negative 0.15, and one of them have very common genetics for your breed, and the other have very uncommon genetics for your breed. When we first got our certificates in standard poodles, we thought that a very low IR meant a dog was very unique for the population, and that is not what IR does. So again, a more negative IR will mean a less inbred dog. A higher IR will mean a more inbred dog. So IR measures heterozygosity. Oh boy, the calculation is not simple. Here it is. So what should you look for? The more negative the IR, again, the more outbred the dog. The genes are more heterozygous or different from one another throughout the genome. It also means that the sire and dam of that particular dog were more different from each other than they were the same. In general, the more positive the IR, the more inbred the dog. the genes are more homozygous or more the same throughout the genome. So that means that the sire and dam of the particular dog you're looking at were more like each other than they were unlike each other. So what's the limitation to IR? And this quote's from a paper that we read in Standard Poodles when we were researching the values that we were given from UC Davis. Internal relatedness is currently one of the most widespread used index, and its main attribute is that allele frequency is incorporated into the measure. However, IR underestimates heterozygosity of individuals carrying rare alleles. So what does that mean in terms of a limitation? IR is useful for populations with very well represented and widespread genetic diversity. However, it's of limited value in populations needing to redistribute the diversity they have maintained after bottlenecks. So again, as we said, a dog can have low IR, but also have very common genetics for their breed. Now here's the examples of breeds that are very well distributed and therefore IR is more important for them. Havanese and miniature poodles. They have maintained their genetic diversity breed-wide, so breeding for a low IR is useful, very useful for them. So here's a well-distributed population, Havanese. And when we say well-distributed, we're talking about allele frequencies. So there are some low represented alleles, but they also have a lot of different alleles in their population, and they're pretty well distributed among the dogs that are sampled. So here you can see their frequencies, and you still have some that are carried by the majority of the population, but there's a lot of them and they're fairly well represented in the breed. So what is a bottlenecked population? What are we talking about when we say that? There are populations that may have many markers at the STRs, but they're not well represented. So in Standard Poodles, we have lots of different STRs in our breed, but the majority are not carried by the majority of dogs. And these populations need to determine if a dog has rare or common alleles. So a population may have 20 markers at one location in the population, but only one or two of those markers are carried by the majority of dogs. Depleted populations have less markers overall than non-depleted. So there are other breeds who don't have lots of markers at each STR. They might only have 4. And those populations really need to try to maintain the diversity that they have, or try to test more and more dogs to see if there's more diversity out there that we're missing. Again, a dog can have the exact same heterozygosity as another, but because it has rarer alleles, it'll have a higher IR. So here's an example of a bottleneck breed, Doberman Pinschers. They have really high numbers of certain types of alleles that are carried by the majority of their population. So 80% of the alleles tested were this particular marker, and here we've got 70% of the alleles tested were this marker. Some examples of breeds that are not well distributed and are bottlenecked and or depleted are Standard Poodles, Akitas, Italian Greyhounds, Doberman Pinschers, really a lot of the breeds that have been tested. So what does IR tell you then? IR is about an individual dog. It means very little in terms of the population. And I'm going to say it over and over again because it's very commonly a misconstrued idea is that IR means that the dog is unique for its population if it's very low. So the more genes that are the same, homozygous, increase the likelihood of recessive mutations being expressed in a dog. So this is why IR is important. So more outbred dogs with a low IR are less likely to have a recessive disorder express or a recessive component disorder. High IR dogs have more likelihood to have genes that are the same and therefore express recessive disease, because a recessive mutation is 2 copies of the same gene. IR does not tell you if your dog has unique genetics in reference to the population. The benefit of IR for litter planning. Litters with low IR ranges have less likelihood to express recessive component diseases. For example, in Standard Poodles, low IR was found to have less risk for sebaceous adenitis. Additionally, in Italian Greyhounds, a low IR was found to reduce and mitigate risk for autoimmune disease. Having low IR in your predicted litter range is very beneficial then. Programs can help you determine the predicted IR ranges for litters, like Better Breed. Is there a time a high IR might be a benefit? This is an interesting concept to consider. So in some instances, a high IR may be of benefit to the population, but perhaps not the individual. Why? As discussed, some populations have bottlenecks and their diverse rare genetics are unequally distributed. So in these instances, a dog with a high IR but very uncommon genetics will be able to produce puppies with low IR and uncommon genetics. So why is that? Because they're unrelated to the majority of the breed and they have a lot of the same genetics. They will be able to, other dogs will be very unrelated to them and they'll very easily produce puppies that are outbred because those other dogs wouldn't have been related to them. We call dogs with a high number of rare and neutral alleles in terms of the allele frequencies of the population outliers. However, that dog, it's not a benefit to that individual because that dog may still carry mutations that are uncommon to the particular population, but it could cause disease in that dog. Now, the idea of genetic diversity is to mitigate and disperse all mutations that are within your breed. All dogs will have mutations of some sort. There is no perfect dog. So the idea is to distribute and dilute any disease genes within your population. So why could it be good to select for uncommon genetics? Genetically diverse populations have better health overall. This has been shown in wild populations. They naturally select for mates that are very different from themselves and they have less autoimmune issues. Depleting the genetics of a population increases the likelihood of fixed diseases, especially if the founders and bottleneck dogs of that population carry genes for disease. So we have seen some breeds that have better health that are bottlenecked, and we believe that's partially because the founders of that breed have had less disease genes than other breeds. But that does not mean that we should still not try to preserve the genetic diversity within those breeds. Zoo populations now are maintained by preserving genetic diversity within the population, and they have lots of different strategies that they follow in order to maintain the population's genetic diversity. And it's well known that when a population's genetic diversity decreases, there's lots of issues that come about. So in summary, IR is informative, but it doesn't tell you everything. What other measurements might help us identify uncommon genetics? So in the upcoming presentation, we will discuss two tools implemented on Betterbred: average genetic relatedness and outlier index. Looking at several measures together can help you plan for healthier litters. Just like we've always weighed multiple aspects of a dog: temperament, structure, drive, other health testing, several measures are needed for genetic planning of litters in most dog breeds. And we definitely do not ever suggest just looking at the genetics. We still need to look at all the other things we have always looked at in our litters. If you have a dog in Betterbred, please update them with their color, location, titles, pedigrees. We hope to offer a new feature soon that will allow for sorting of potential breeding mates. Questions? Email support@betterbred.com, or you can ask in your specific breed group. Thank you for watching.